So I'm going to look at PCR first because we essentially need to do it in order to do gel electrophoresis. PCR is like a genetic photocopy machine. It's used to make millions of copies of a specific segment of DNA. It is slightly different from recombinant DNA, which we just looked at, and it's the preferable method in the modern age because it is generally faster and does not require as many materials. Again though, it does use an enzyme, and this is DNA polymerase, which acts like a molecular builder joining individual nucleotides together to form DNA strands. This is the exact same polymerase that is responsible for DNA replication. That's why, in essence, PCR is sort of like artificial DNA replication. One of the cool things as well about PCR is the enzyme it uses, which is another enzyme it uses, which is Taq polymerase. This enzyme is really special because it doesn't fall apart at high temperatures because it's taken from hot springs. This quality is really crucial for how PCR plays out. So I'm going to go over the steps involved in it and you'll begin to see why. The first step is denaturation. In this step, the DNA is heated up, which causes the double-stranded DNA to unzip and separate into two single strands. You can think of it like unzipping a jacket. It breaks the hydrogen bonds holding the strands together. If you take chemistry, you'll know that hydrogen bonds are a lot weaker than covalent bonds. So it makes sense, essentially, that the hydrogen bonds are untied before when you increase it to a certain temperature. And that's why we need to use Taq polymerase, because if we're putting it up to these high temperatures, Taq polymerase would fall apart and denature as well. But it doesn't, thanks to its quality. Next comes annealing. The temperature is lowered to let small pieces of DNA called primers attach to the complementary sequences of the single strand. And as you might know from DNA replication, primers are like the starting points that signal where the copying process should begin. Again, you would have learnt about that in DNA replication. And then the final step is extension. The temperature goes up again to the right temperature for Taq polymerase to get to work. Taq polymerase is, I probably should have mentioned this earlier, Taq polymerase is a type of DNA polymerase. So it starts building new DNA strands from the original ones using the free-floating nucleotides in the mix. These three steps, denaturation, annealing, and extension, just make up one cycle of PCR. So scientists actually repeat this cycle over and over, like hitting the copying button on a printer to make millions of copies of the target DNA segment. It's fast, efficient, and can produce enough DNA to study even tiny amounts found at a crime scene or in ancient fossils. Let me think. Imagine you run this cycle 30 times. If you started with one DNA segment, you would be left with 2 to the power of 30 segments, or over 1 billion. Well, you can see why it might be useful for replicating small amounts of DNA, but why do we care about PCR? Well, it's super handy in forensic for analyzing small amounts of DNA from crime scenes, but it's also used in medicine to diagnose diseases by detecting infections or genetic disorders early on. Scientists even use PCR to try and extract and amplify DNA from ancient fossils or extinct species, which could tell us loads about our planet's history and evolution, which ties nicely into our next topic actually. So there's actually a lot of interesting applications for PCR, but oftentimes, like in crime scenes, it's pretty difficult to do anything with a bunch of DNA. You need a tool to analyze it.